Figure number 2 shows a section through a leaf. A leaf model is given here. The parasite layers are given. This question is from the specialized cells, plant tissues, and photosynthesis. 2.1 is an A1 question. Give one way the palisade layer is adopted for photosynthesis. If you look here, the palisade layer, they are closely packed to each other. They are present near the top of the leaf. On the other hand, they have huge quantity of chloroplast which helps to do more photosynthesis. Let's see the model answer. They contain a large amount of chloroplast or chlorophyll. They are closely packed and they are near to the top of the leaf, so they can absorb more energy from sunlight to do photosynthesis. 2.2 is an AO1 question, which is a knowledge based question, and this question is from plant tissues and organs. Gases pass into and out of the leaf through the small pores in the surface of the leaf. What are the small pores labeled X called? These small pores or holes are present on the lower side of the leaf. So these small holes or pores are called stomata. Sometimes the students make a mistake. They write it as a God cell. God cells are actually present around stomata. These are God cells, but these holes are called stomata. 2.3 is an AO2 question where the students need to apply their knowledge, and this question is related to practical activity number 1 in combined science, and this question is from cell structure and microscopy. A student viewed a section of a leaf using a microscope. The student measured the length of one of the palisade cells. The cell image measured 28 mm in length when viewed at a magnification of 400 times. Calculate the real length of the palisade cell in millimeters. We are already given the size of the image 28 mm or the image length. The magnification is already given, that is 400. We need to find the real size or real length. So image length is coming here. The magnification is used here. Therefore, the real length is 28 over 400. And that is 0.07 millimeters. But the second part of the question is we need to convert our answer from millimeters into micrometers. And the unit conversion is already mentioned. If you want to convert from millimeters to micrometers, we need to time our value by 1000. The answer is 70 micrometers. Two point four is an AO1 question. Carbon dioxide can move into and out of the cells. What is the process by which carbon dioxide can move into and out of the cell? This question is from the topic of diffusion. The answer is diffusion is a process through which the carbon dioxide can move into and out of the cells. Osmosis is related to the diffusion of water, and active transport is related for the movement of ions. So these are incorrect options. Figure number 3 shows a diagram of 4 cells: cell A, cell B, cell C, and cell number D. Each cell is surrounded by carbon dioxide molecules. So all these are carbon dioxide molecules. Two point five is a combination of AO2 and AO3 questions. Which cell will carbon dioxide move into at the fastest rate? If you see here, which cell has highest concentration difference? The concentration difference will decide the movement of the molecule. If you see the cell number A, there is more carbon dioxide is present outside the cell than inside the cell. Therefore, the carbon dioxide will move from outside to inside with the higher rate. If you see in cell number C, there is almost equal concentration is present inside and outside. Therefore, the rate of diffusion is very slow. In cell number D, the chances are the movement of the carbon dioxide is from inside of the cell to outside the cell. Whereas in cell number B, it looks there is an equal amount of movement of carbon dioxide from outside to inside and from inside to outside. The fastest rate of carbon dioxide to move into the cell is cell number A. So the correct option is cell number A. The student need to give the reason. The concentration of carbon dioxide is higher outside the cell A than inside. Therefore, the carbon dioxide gas moves fastly into the cell A. 2.6 is an AO1 question, which is a knowledge-based question, but it is related to practical activity number 5, which is about the rate of photosynthesis. In our diagram, we have pond weed, we have the timer. The bubbles of gas are formed here, we can see in the diagram. The student investigated the effect of different colors of light on the rate of photosynthesis. So the key information is colors of light. The apparatus is shown here in figure number 4. The student placed the apparatus in blue light, then in green light, and then in red light. The student measured the rate of photosynthesis in each color of light. Question is, which two measurements should the student make to calculate the rate of photosynthesis? Student need to calculate the number of bubbles produced in the fixed time. The time can be 5 minutes, 10 minutes, or 15 minutes for each color of light. The model answer is the volume of the gas collected in the test tube, or count the number of bubbles formed there. And the student need to use the time for 5 minutes, 10 minutes, or 15 minutes for each color. So these two measurements should the student make to calculate the rate of photosynthesis. 2.7 is an AO3 question, which is from the practical activity number 5. And this question is from the rate of photosynthesis. Give two variables The student should keep the same in this investigation. The variables which are kept constant, they are called controlled variables. The control variables in this question are the variables which are kept constant in the investigation are the temperature of the water. And second is the distance of the light from the pond weed or the concentration of carbon dioxide in the water also. So these are the variables which must be kept constant or same throughout the investigation to see the effect of light colors on the rate of photosynthesis. Let's see the model answer. The variables which are same throughout the investigation are temperature of the water, are the distance of light from the pond weed. Even the student can write the concentration of carbon dioxide in the water. Table number 2 shows the colors and the rate of photosynthesis in arbitrary units. We can see that when it is blue light, the rate of photosynthesis is very high, the green one is very slow, and the red one is 8. 2.8 is an AO2 question. The students need to apply the knowledge. Complete the figure number 5. You need to label the y-axis, use of suitable scale, plot the graph from table number 2 as bar charts, and label each bar. We can see from here in figure number 5, I put the table next to it. I labeled my x-axis. I put the scale on my y-axis from 0 to 10. My y-axis are rate of photosynthesis in arbitrary units. From here, from the table, you can see that the blue is 9. So we can draw the blue graph here and we can label it blue. For the green one is 1. So this is my green one. The rate of photosynthesis, which is green color, is 1. So it is reflecting 1. And for the color of light for the red, it is 8 according to the scale. So we put it 8 here and we are drawing a bar chart. 2.9 is an AO2 question. Look at the table number 2. Which color of light should be used to grow plants in a greenhouse? Tick one box. We can see that blue, green, and red. The rate of photosynthesis is higher in blue color. So, in the greenhouse, the light which should be used is a blue light.